Yeah, okay, we'll start first. So, hello everybody, welcome to today's webinar. We'll be talking about respiration today. So this is your learning outcomes. You can see here we have a space here, but that's because, that's for photosynthesis. So I just blanked out the photosynthesis part and just focus on respiration here. So the first thing that you guys have to be able to do is to identify components of your mitochondria in drawings, photo micrographs and electron micrographs. You have to talk about, sorry, not this. You have to talk about the process of glycolysis, the location, raw materials needed, products formed, and you have to know link reaction Krebs cycle, again, location, raw materials, and products. And then OXPHOS, the roles of oxygen, electron transport chain in both aerobic, in aerobic respiration. Then we'll talk about anaerobic respiration and how it produces only a small yield of ATP. And we'll talk about anaerobic respiration in yeast and in animals or mammalian muscle tissue. We also talk about the significance of ethanol in yeast, lactate in mammals, and the regeneration of NAD. Okay, then this one here is more of like practical. And then we also have chemiosmosis, which is very, very important in respiration. So an outline of today's lesson will be covering content, how to effectively memorize all the different compounds, and how you get this, how you see respiration as a small picture and how you see it in the bigger picture in your in your biology syllabus. We also talk about some links between respiration and other topics as well. I'll give you a typical respiration question and a tough respiration question. Again, inter and intra-topic links. And if you have any questions, you can type them in the chat at any point in time, or you can raise your hand. I will try my best to look up for people who raise their hands. But we'll have breaks to the way. So if you have any questions, any point in time, you can ask in the break. Preface. So different schools have different keywords and different terms. So one example for today will be later we will talk about Krebs cycle. Some schools don't cover the names of the compounds, but my school did. So I'll be teaching you guys the names of the compounds. But if your school doesn't teach you, you don't have to go and memorize them. I'll just take it as a 4-carbon, 5-carbon, 6-carbon compound. Yeah. So today we'll be following RI syllabus. Okay, so a lot of you guys, quite a few of you guys were talking about how to remember all the different compounds here. So these ones here are glycolysis. This is link reaction. And this is your Krebs cycle. So how do you remember all these things? Okay, so I'll show you a quick TikTok video made by us. And yeah, okay.
Struggling with respiration? Try this.
Okay, so the how I remember the compounds in respiration is my teacher taught me this. It sounds a little weird at the start, but then after you say it a lot of times, it actually stays in your head a lot. Okay, so it's GFG 13 GPA OC alpha. Okay, so this is how I remember it. So I keep the first time he told us, I was like, there's no way I'm going to remember all these things. But then the more you say it, and if you break it down to these different parts, you can remember it quite easily. Yeah. So so G is glucose. Then you have fructose 1,6 bisphosphate, glyceraldehyde 3-phosphate, 1,3 bisphosphoglycerate, glycerate phosphate, pyruvate, acetyl CoA, oxaloacetate, citrate, and alpha ketoglutarate. Okay, so yeah, that is how I remember it, and it's unconventional. It doesn't sound like it'll help, but the more you say it, the more it actually helps. The more it stays in your head. Okay, so we'll start with glycolysis first. So glycolysis, we have our on the right side is your number of carbons. On your left side is the molecule. Okay, in blue is what is needed, is what is produced. So the raw materials and the products, and the ones in purple are the names of the reaction that occurs. Okay, so the first thing is your glucose becoming fructose 1,6-bisphosphate. So if you look at the ones on the right, the compound, from a 6 carbon, it stays a 6 carbon, but suddenly there are 2 phosphate groups there. Okay, that's why there's -2 ATP here. And then, so this process phosphorylation. Next one, we have lysis. So fructose 1,6-bisphosphate lyses to form 2 times glyceraldehyde 3-phosphate. So one thing that a lot of people forget is that after lysis, they forget to add the 2 times here. Okay, so they only leave it as 1. So when they calculate the number of products formed, it gets a little messy. So just remember that it's 2 times of glyceraldehyde 3-phosphate. So if you pay attention to the story on the right, 6 carbon becomes 3 carbons, and each of the 3 carbons has your phosphate group. Then 2 times glyceraldehyde 3-phosphate undergoes oxidation. Products formed are 2 NADH. Okay, and then you get your 2 times 1,3-bisphosphoglycerate. So if you see the story to the right again, you have your 3 carbon compounds, and then now each of the 3 carbon compounds has 2 times phosphate groups. Okay, then after that you get your glycerate phosphate via substrate level phosphorylation, and you get 2 times ATP produced. And after glycerate phosphate, you also glycerate phosphate undergoing substrate level phosphorylation, you get 2 times pyruvate, and again 2 times ATP is produced. So if you want to, so I've combined the products at the bottom for all the reactions. So later for link reaction and for Krebs cycles as well. So if you want to, you can take a screenshot of the glycolysis portion. Okay, so it might seem daunting to have to remember all these different molecules, but just use the GFG13 GPA OC alpha. Okay, the more you say it to yourself, the easier it gets to remember. So we'll first talk about anaerobic respiration. So if O2 is absent, we have to undergo anaerobic respiration. So there are 2 types of anaerobic respiration. We have alcohol fermentation, we have lactic acid fermentation. Alcohol fermentation occurs in your yeast and in some bacteria. So this is the cycle, sort of how the entire process works. So first we start off with glucose, and it undergoes glycolysis, and then in the end it forms your pyruvate. So your pyruvate here. So glucose to here, this is your glycolysis. So 2 ADP goes in and 2 ATP comes out. We're talking about net here. Okay, so net 2 ATP, and after glycolysis occurs, pyruvate decarboxylase, the enzyme, so all the enzymes are labeled in green. Pyruvate decarboxylase is involved, and it converts your 2 times pyruvate to 2 times ethanol, and then because pyruvate is a 3 carbon compound and ethanol is a 2 carbon compound, you have to lose carbons, and the only way carbons are lost in respiration is via CO2. Okay, so because 2 times pyruvate and 2 times ethanol, therefore you have 2 CO2 molecules, and then from your ethanol to your ethanol, you have to you use alcohol dehydrogenase to get 2 times ethanol, and then your 2 NADH here becomes 2 NAD+. So this brings in the idea of the regeneration of NAD, and we'll talk about why that is important later on. Okay, we'll move on to lactic acid fermentation here first. If you want to take a screenshot, you guys can do this by yourself, which will make it easier for you to remember. Okay, yeah. This is what I did, and then, I mean, it helped me remember all these different compounds. Okay, so talk about lactic acid fermentation. So lactic acid fermentation, again, is in your mammalian animal tissue. So glucose becomes pyruvate. Same story here. It's because of glycolysis. And after you form your pyruvate, the difference here is that you get lactate. Okay, lactate is not like ethanol. It's not like ethanol. It's not like ethanol. It's a 3-carbon compound. Therefore, there's no loss of there's no CO2 produced. Therefore, no CO2 here. Okay, and then again, because the function of anaerobic respiration is to reproduce or to produce again your NAD+, to regenerate your NAD+. That's why your NADH gets oxidized to your NAD+. Okay, now we talk about the functions of your anaerobic respiration. Okay, so the first one is to produce a small yield of energy, and this is 2 ATP per glucose molecule that enters. And the second one is to regenerate your NAD+ from your NADH. And why you need to regenerate your NAD+ from NADH, okay, why you need to regenerate your NAD+ from NADH is because you need your NAD+ to get reduced. That's why there's that upper arrow here, and that's why there's that lower arrow here. Okay, so the upper arrow, your NAD+ has to get reduced to NADH to convert glucose to pyruvate. But your NADH is used to reduce ethanol to ethanol. Yeah, because NADH gets oxidized to NAD+, which means it reduces ethanol to ethanol. Okay? Yeah, you guys can, if you want to, take a screenshot. Okay, and a lot of people forget to study the functions of. A lot of people forget to study the functions of anaerobic respiration. So it's to regenerate your NAD+ from NADH so that you can continue producing a small yield of energy, and the small yield is 2 ATP. So it's very important to remember 2 ATP, and then to remember the other number versus aerobic. Okay, so you have to remember how many ATP are produced in aerobic respiration, which we will cover now. Okay, so the story just now was if O2 was absent. If O2 was absent, but now what if O2 is present? Okay, so we go through aerobic respiration. So we first have to go through link reaction, and link reaction is the easiest one here. Okay, so it undergoes oxidative decarboxylation, and the products formed are 2 NADH and 2 CO2. Okay, again, 2 because you have to go through link reaction 2 times because you have 2 molecules of pyruvate. Okay, because previously you have 2 molecules of pyruvate, therefore here you also have 2 molecules of pyruvate. So again, your CO2 comes out here because pyruvate in this part is 2 times a 3-carbon compound, and your acetyl-CoA is 2 times a 2-carbon compound. So from a 3-carbon to 2-carbon, the only way carbon can be lost is via CO. Is when it is in the form of carbon dioxide molecule. Next is your Krebs cycle. So this is where there's contention, but not really contention. There's difference in content across schools. Okay, so okay, let me write this down. So oxaloacetate can be and is referred to as a 4-carbon compound in some people's syllabus. Citrate is referred to as a 5-carbon compound or 5-carbon intermediate. Alpha ketoglutarate is referred to as 4-carbon compound or 4-carbon intermediate. Okay, so yeah, again, these are represented by the boxes beside their names. Okay, so we start the story off with oxaloacetate. Oxaloacetate is a 4-carbon compound. It exists there already. It combines with your acetyl-CoA, which was again produced from your link reaction from before. So it combines with your acetyl-CoA, CoA leaves, coenzyme A leaves. It forms your citrate. Citrate is a 5-carbon compound. Okay. Sorry, citrate is a 6-carbon compound. What am I? Citrate is a 6-carbon compound because this is 4 carbons, this is 2 carbons, therefore equals to 6 carbons, which is citrate. Okay, so your citrate then undergoes oxidative decarboxylation, and then it gets, undergoes oxidative decarboxylation, and whenever it undergoes oxidative decarboxylation, oxidative, therefore you have NADH. Decarboxylation, therefore you have CO2. Okay, so yeah, so whenever there's oxidative decarboxylation, that means that both CO2 and NADH are produced. Then from citrate to alpha ketoglutarate, which is a 5-carbon compound, now the alpha ketoglutarate has to undergo oxidative decarboxylation to form oxaloacetate. Oxaloacetate was the compound we started with just now. It's a 4-carbon compound. This is a 5-carbon compound. There's only one way carbon can be lost, therefore there is CO2, and this is decarboxylation. Oxidative decarboxylation, therefore you have your NADH as well. But it's different here because you also have your FADH2. So you have your 1 times FADH2, 2 times NADH, and there's substrate level phosphorylation as well. Therefore, there is 1 ATP produced here. Okay, so the products are very important to remember, but it's very easy to remember as well. Because the only thing you have to remember is, here, which is your this one here and your this one here. You only have these 2 things that you have to remember as products. And of course, coenzyme A here, but that's not very important. So you just remember that as long as you lose a carbon, it has to come out as CO2. So that's how you can help to remember your compounds. And you have to remember that you have FADH2 as well. So it has to come from somewhere, and it doesn't come from the first reaction, it comes from the second reaction. That's how I always remembered it. FADH2 has to come out from somewhere, but because there's 2 here, therefore it comes out the second time. It doesn't come out the first time. Okay, and then this 2 also reminds me that there's 2 NADH. Okay, that's how I remembered it. Okay, FADH2, the 2 there means that it's the second reaction, and it means that there are 2 NADH produced with it. Okay, so the products here will be your 2 times ATP, 4 times CO2, 2 times FADH2, and 6 times NADH. So why are these numbers then different from the ones that I wrote above? Because it's from 2 cycles. Okay, so 2 cycles. Why? Because we are taking relative to 1 glucose molecule. So 1 glucose molecule after the link reaction will produce 2 times acetyl-CoA. Will produce 2 times acetyl-CoA, and therefore it will undergo Krebs cycle twice per glucose molecule. Okay, next is your, okay, before we start on oxidative phosphorylation, we'll have a 3-minute break. Okay, we have a 3-minute break. If anyone has any questions, they can unmute themselves and we can ask them right now. Okay, a 3-minute break for you to digest glycolysis, link reaction, Krebs cycle, and oxidative phosphorylation. Okay, this is it without any drawings or anything. Does anyone have any questions? If you have any questions, just ask them. And if you don't want to like voice them out, you can just, oh, okay. Is substrate level phosphorylation just the addition of phosphate groups? Addition of phosphate groups to ATP to form ATP. Yes. Yes, Keegan, it is. Substrate level phosphorylation. No problems. No problems. Substrate level phosphorylation. Yeah. Okay, so another thing that I actually forgot to cover just now was the locations at which these things are happening. Okay, because in your learning outcome, you have to remember where the locations, you have to know where the locations are. So your glycolysis occurs in your cytosol. Cytosol is your cytoplasm. Your link reaction and Krebs cycle both occur in your matrix. Okay, sorry, the glycolysis one is here. In your cytosol. Glycolysis in your cytosol. Link reaction and Krebs cycle both in matrix. Okay, so let's talk about the basis of oxidative phosphorylation first. Okay, the basis of oxidative phosphorylation is that your NAD plus and your FAD molecules are your mobile electron carriers. Okay, they carry your electrons and they can move, and that's why they are mobile electron carriers. Contrast your mobile electron carriers with your electron carriers in your ETC, your electron transport chain. So electron carriers in your electron transport chain cannot move, and therefore they're not called mobile electron carriers. Okay, so your NAD plus and your FADs both carry your electrons, your high energy electrons, to your electron carriers in your electron transport chain. So this is a keyword. Okay, high energy electrons. Always use high energy before the words electrons for respiration, for biology. And why you have to emphasize the high energy is because later we talk about the energy coupled thing that I was talking about just now. Okay, so each electron carrier in your ETC has an energy level lower than the one preceding it. What this means is, okay, if we look here, can I draw on this? Okay, so if you see this one here, okay, the first one that I pointed to, and this one here, this is the second, this is the first. The energy level is like this, okay? And why it has a lower energy level is because it gets increasingly electronegative. Your electron carriers, your first, second, and third, let's talk about it, okay? Your first, second, and third, energy level decreases. Energy decreases in this direction. Electronegativity increases. Electronegativity decreases in that direction, okay? So it gets increasingly electronegative, but the energy level decreases, okay? So that's what it means. That's the basis of oxidative phosphorylation. If we talk about the actual oxidative phosphorylation, which is the actual process of oxidative phosphorylation, your first step is that your NADH molecules, okay, that were generated from before, which is during glycolysis, link, and Krebs cycle, okay, your NADH molecules generated from here all get donated to your electron carriers in your electron transport chain, as you see in the GIF there. Okay, so some things that I want to talk about here is NADH. So some schools don't talk about NADH; they refer to it as reduced NAD. Both are perfectly okay. And then some of you might ask, why am I only talking about NADH molecules, and why am I not talking about FADH2 molecules in this step 1? Because your NADH molecules donate to your first electron carrier of your electron transport chain, but for your FADH2, they transport it to electron carriers that are halfway, they are in the middle of the electron transport chain. They don't donate it to the first electron carrier in your electron transport chain. Okay, so the story continues. If your high energy electrons travel down your increasingly electronegative electron carriers, okay, the energy is released. So energy is released because before we talked about energy level being lower than the one preceding it. So the first one, let's say it has 1,000 joules, just an arbitrary unit, 1,000 arbitrary units of energy, okay? The second one will have less than 1,000, let's say 900. So that 100 joules of energy, assuming ceteris paribus, okay? Assuming ceteris paribus, your 100 arbitrary units drives your protons across your membrane into your intermembrane space, okay? Like in this GIF here. Okay, so as they travel down your increasingly electronegative electron carriers, energy is released. That energy released is coupled to the pumping of protons of your H plus ions from your matrix into your intermembrane space. Okay, so far, what has happened is electrons are donated. Electrons move. That movement of electrons releases energy. That energy released is used to pump protons from your matrix into your intermembrane space. And the thing here to be comfortable with is you have to be comfortable with the interchanging terms H plus ions, protons. Okay, they can be referred to as both. It depends on your school, and in Cambridge, they use it interchangeably as well. So in your answers, you can use them interchangeably as well. So the fourth step is, so the concentration of your protons now in your intermembrane space increases. Okay, if you look at the GIF, again, before there were very few, but in this picture, before there were none. But now as your NADH and FADH2 donate their protons, now there is an increase in concentration. Now there are more H plus ions in your intermembrane space. So the concentration of your protons increases in your intermembrane space, and this builds up a proton motive force across your cristae. Okay, so what is a proton motive force? Your proton motive force is just potential energy. Okay, all it is potential energy. Potential energy stored in the form of a proton gradient. Because just now, okay, let's say this is the cristae. Then this is your matrix. This is your intermembranal space. So your H plus moves from here, there. So we have more H plus here. So that's why there is a gradient, because the IMS has greater concentrations of protons than your matrix does. And that proton gradient has energy stored in the form of potential energy, which is your proton motive force. Okay, so as your H plus now diffuses down its concentration gradient, which is from your intermembrane space back to your matrix. Okay, so as it diffuses down its concentration gradient through your ATP synthase, ADP is phosphorylated to ATP. So that's how ATP is formed. Okay, that's how ATP is formed in oxidative phosphorylation. And then the last part is that O2 acts as your final electron acceptor. Acts as your final electron acceptor. And if you contrast this with your photosynthesis, right? If you contrast this with your photosynthesis, you see how these two processes have different molecules acting as the final electron acceptor. So if you look at the 2020 A-level bio paper, I want to say 3, the essay, the one with the essay, right? They talked about the differences between respiration and photosynthesis. So a lot of people wrote this as one of the contrasting points, as one of the reasons why respiration is different from photosynthesis. And it's very important for you guys to remember it as well. Even though it will not come out as an essay question, it can come out as a short answer question. Okay, so contrast it with your photosynthesis, and then you get a clearer picture, and you can better differentiate in your mind respiration and photosynthesis, because a lot of times we get confused between the names of the compounds and the entire process because they are quite similar and we study them very close to each other. Okay, so in this case, O2 in respiration is your final electron acceptor, and this is the equation that is used. Okay, this is the equation that occurs. So O2 is your final electron acceptor, combines one oxygen atom combines with 2 electrons and 2 protons, and then it forms your water molecule. And there are functions of this water molecule, also known as metabolic water. Desert animals use them a lot to get sources of water. Okay, so as H plus diffuses down its concentration gradient, and how ADP phosphorylates to become ATP, right? That is an example of chemiosmosis. So another question that Cambridge loves to ask is, please define chemiosmosis. So chemiosmosis is just energy stored in the form of proton gradient across a membrane. And in this case, the membrane is your cristae. You can see in the picture, in the GIF. Okay, across a membrane, and it's used to drive cellular work. So just break this down. Simple, just break it down, and you'll be able to remember what chemiosmosis' definition is. Okay, so energy stored in the form of a proton gradient, which was your proton motive force, and then it is generated across a membrane, and the energy is used to drive cellular work. Okay, so that's the definition of chemiosmosis. And the example of chemiosmosis here is point number 5, which is the diffusion of H plus down its concentration gradient through the enzyme known as ATP synthase, and then that causes ADP to be phosphorylated to ATP. Okay, so that's oxidative phosphorylation. Is something that is daunting to some people because it is very difficult to understand the first time you learn it. The first time you come across it, it's difficult to understand. Okay, so the answer here is substrate level phosphorylation is not oxidative phosphorylation. Substrate level phosphorylation is the direct phosphorylating of ADP with phosphate and energy provided from a coupled reaction. Substrate level phosphorylation only occurs if there is a reaction that releases sufficient energy to allow the direct phosphorylation of ADP. Oxidative phosphorylation is when ATP is generated from the oxidation of NADH and FADH2 and the subsequent transfer of electrons and pumping of protons. Yeah, no, okay, so they won't really ask this because I've not had a question in the prelim papers or in the TYF. They haven't asked this, but substrate level phosphorylation, usually we don't refer to it, we don't talk about it in oxidative phosphorylation. We talk about it in your everything prior to oxidative phosphorylation. Okay, because yeah, we don't refer to the generation of ATP from ADP in oxidative phosphorylation as substrate level phosphorylation, but we refer to it as oxidative phosphorylation. SLP is used for anything prior to OXPHOS. Okay, it's used in your Krebs cycle. Is used in your Krebs cycle and is used in your glycolysis. Okay, next question is, how is it that down the ETC it's increasingly electronegative? Well, okay, you don't have to know why it is increasingly electronegative. Okay, you don't have to know why, you just have to know that it is. I personally never cared why it is increasingly electronegative. I have honestly not I've never asked myself why. Okay, why? What's the question now? Why is okay? We can do this because today we have less people, so we do this. Okay, why are electron carriers increasingly show negative? Yeah, okay, actually, yeah, isn't it just because if it's more okay, electronegative definition, right? A tendency to acquire electrons, right? So if it is increasingly electronegative down the electron transport chain, that's why from one carrier to the next carrier, which is more electronegative, can your electrons flow. Does that make sense? If it's more electronegative, it means that it has a higher tendency to acquire the electron from the previous electron carrier. So if there's no difference in electronegativity, then your electrons won't flow down your ETC. Okay? Yeah. We didn't have to, just, yeah. Okay? I hope that cleared it up for you, fluffy clouds. And I hope I answered your question, Keegan. Yeah. Okay, so now that we're done with that, let's go on to the importance of. Okay, so a lot of questions and a lot of papers like to ask the importance of these things. So the importance of, let's talk about oxygen first. The importance of oxygen is, of course, that it is your final electron acceptor. If this is a 2-mark question, as long as you write final electron acceptor, you're getting 1 mark. Okay, it's that simple. Oxygen is your final electron acceptor. It allows your mobile electron carriers, and of course, the mobile electron carriers here refer to our NAD and FAD, whether you want to put the H2 and H. Okay, so these are your mobile electron carriers. It allows for them to continue donating their electrons to your electron carriers in your ETC, and therefore it allows OXPHOS to continue. And if OXPHOS can continue, ATP can be generated. So the importance of oxygen, first mark is your final electron acceptor. Second mark goes to everything else, which is it allows it to continue donating. Okay, that's half a mark. Continue donating, allowing OXPHOS to continue. That's another half mark, and then ATP is generated. There were no marks, but it's inferred from OXPHOS being able to continue. Okay. Then the importance of your ETC. So the first importance of ETC is that it aids in the generation of your proton motive force. So your proton motive force is, of course, what drives chemiosmosis afterwards, right? So it aids in the generation of proton motive force to produce ATP. If you want to elaborate further, to allow for chemiosmosis, and therefore ATP can be produced. Second importance is it regenerates your NAD+, it regenerates your FAD, therefore your link reaction, your glycolysis, your Krebs cycle can continue. If NAD+ is not regenerated, if FAD is not regenerated, these 3 processes cannot continue to occur. And if these 3 processes cannot continue to occur, OXPHOS cannot occur, and therefore very few ATP, or specifically 2 ATP, will be produced. So if NAD+ and FAD is not regenerated, that's usually because O2 is not present. Because O2 is not present, OXPHOS cannot occur. If OXPHOS cannot occur, NAD+, FAD cannot be regenerated. Therefore, link, glycolysis, Krebs cannot occur. That's why we go the other route, right? Because remember, there are 2 routes to respiration. After glycolysis, there is your anaerobic respiration, okay? And depending on which organism it is, we'll talk about two different things. Okay, two different types of anaerobic respiration. Okay, so yeah, someone asked, how do you remember the what's this called? Molecules involved in your reactions. So alcohol fermentation. So what is alcohol? Alcohol is ethanol. That's how you remember. Ethanol is a type of alcohol, therefore the ethanol thing is involved in alcohol fermentation. Lactic acid is lactate. Okay, if you want to know why lactic acid is lactate, it's because lactic acid, it doesn't stay as lactic acid in the body. It is lactate. Just like your citrate here is citric acid as well, but it's citrate here. If you want to know, you can go and you can stay back after the lesson and I'll explain it to you because it's quite, yeah, okay. But just know that there's a cool thing that happens here. Okay, citrate and lactate. That's why it's not lactic acid, because lactate. Okay, so yeah, I have just completed the entire respiration syllabus. Yeah. So remember everything about it is your GFG 13 GPA. Just remember that and you'll be set. Okay, just remember those these words, these alphabets, and then you'll be able to remember all the molecules there. Okay, and then when you study, you just have to write down beside it. Okay, so the big part in glycolysis is lysis. Okay, lysis is when your 6 carbon breaks into your 2 times 3 carbon compounds. That's how you get 3 carbon compounds, and acetyl-CoA is your 2 carbon compound. Krebs cycle is math, just math. Okay, 4 plus 2 you get 6. 6 minus 1 you get 5. 5 minus 1 you get 4. Why minus 1 is just because that's how Krebs cycle works. Okay, so just remember it's plus minus minus. So 4 plus 2 6, minus 1 5, minus 1 4. Okay, we'll move on to the typical respiration question. Okay, we'll go back to the MCQ. Okay, if you guys want to do it later, but we'll go through the questions, the open-ended ones first. Okay, this is your typical easy question. Okay, it's a easy question. Okay, so I think we'll start going through the question. Is that okay? Or does anyone need more time? If you need more time, you can just type it in the chat. Okay, so we'll go through, okay? Okay, so the first part of the question is this. Okay, it's 4 marks. So 4 marks means you need your 2 distinct points. So yeah, okay, it just means you need your 2 distinct points. So how I structured my answers was, I first talked about first looked at the word results. So results and table mean that I have to quote the results given in the table. And support chemiosmotic theory. Chemiosmotic theory is here at the start. Synthesis of ATP using proton gradient across the membrane, right? So I just had to link it back to the chemiosmotic theory. So the first comparison I took was comparing between 1 and 2. So comparing between 1 and 2, ATP was produced in 1. ATP produced in 1, but not in this 2. Okay, so that's my quote, and that's my that's the difference. Okay, so now I link it and I explain why. So I say that in the presence of oxygen, and then oxygen is the final electron acceptor, electron transfer along the ATC is allowed. Okay, proton gradient can be established, ATP can be produced. Okay, so I took 1 and 2, and the only difference between 1 and 2 is that oxygen is present in 1, oxygen is not present in 2. So let me think about aerobic respiration. Why is oxygen needed? Because it is the final electron acceptor. If it's not present, it cannot accept electrons. No electron transfer can occur. No proton gradient can be established. No ATP can be produced. Okay, second point, comparing 3 and 4. If you compare 3 and 4, the only difference here is your concentration of protons. 3 has low concentration, 4 has high concentration. So if we compare 3 and 4, let's quote first. Compare 3 and 4. ATP produced in 4, not in 3. So let's try to link it back to the chemiosmotic theory, and let's explain why. So the high concentration of protons allows for your proton gradient to be established across your inner mitochondrial membrane, across your cristae, and the diffusion of your protons from your intermembranal space into your matrix, okay, allows for ATP to be synthesized in 4, but not in 3. Okay, the high concentration of protons allows for a proton gradient to be established across your inner mitochondrial membrane. Okay. Okay, I'll let you guys have time to digest that. Okay. Okay, let's move on. So you account for the consequences to a mitochondrion if the water potential of the liquid in the dish is less negative than the water potential of the mitochondrial matrix. So less negative means that the water potential of the dish is greater than the water potential of the matrix. Okay, so just this question is just osmosis. Okay, so water enters your matrix by osmosis, and of course down the water potential gradient, and then your membrane eventually ruptures and your mitochondrion bursts. It's a very simple question. Okay, next, explain. Okay, and then aerobic respiration, and it's specifically the role of your matrix. So your first point is that the matrix is the site of your link reaction. Oops. Your link reaction is your site, your matrix is your site for your link reaction and your Krebs cycle. Okay, oxidative decarboxylation forms acetyl-CoA from pyruvate, which is needed in your Krebs cycle. So matrix allows for link reaction to occur. Link reaction allows for Krebs cycle to occur. Okay, then continue on the story. In your Krebs cycle, you have your NADH, you have your FADH2 that are produced, which is needed for oxidative phosphorylation, which is the process that produces the most ATP in aerobic respiration. Okay, so matrix is the site where link and Krebs occurs. So explain. Link, oxidative decarboxylation occurs, therefore acetyl-CoA can be produced. Krebs produces NADH and FADH2, which is needed for OXPHOS. OXPHOS generates the most amount of ATP in aerobic respiration. Okay, let's move on to the tough question. Okay, so this is a tougher paper, a tougher question. Okay, so let's go through the question. So, okay, the big clue here is that mitochondria is not the cell. Okay, mitochondria is an organelle, as we all know. But so that's why the preamble of a question is actually very important, because Most of the time, we just brush through it to get a rough idea of what the question means. But in this case, it is very important because it talked about mitochondria. It didn't talk about a cell. So what's different between a mitochondria and a cell? A cell has a cytoplasm, whereas the mitochondria does not, right? So first question is, explain why glucose cannot be the respiratory substrate that was added. So explain means reason plus explanation. Okay, so the reason why it cannot be is because your cytoplasm / cytosol is not present in your suspension. Therefore, glycolysis cannot occur. So the location of glycolysis is your cytosol, and that's why glucose cannot be added, because glucose is the beginning product of your glycolysis. Therefore, glucose cannot become pyruvate because your cytosol is not present, and because pyruvate is not generated, your subsequent link reaction Krebs cycle oxphos cannot occur. Therefore, there won't be a dip when ADP is added at time A and B. Okay, so the explanation here is because glucose cannot be broken down and it cannot be utilized in your mitochondria. There's another answer, sorry. Your first answer is here, second answer is here. Okay? Two separate answers. Okay. Then the explanation part is because, the explanation part here is because your pyruvate is needed. Okay, as a substrate for link Krebs oxphos okay, it's because pyruvate is needed. That is the explanation as to why glucose is not the respiratory substrate that was added. Okay, so the first two, what I labeled 1 and 2, that is the first part of your answer. Either one of them can work. The second part of your answer is that pyruvate is needed for LK and oxphos. That's why glucose could not have been the respiratory substrate. Okay, I'm assuming there will be questions. Are there any questions? This is quite a tricky question because no one would have thought that that was the answer. Okay, if there are questions, just type them in the chat. Okay, no one's typing. Okay, part 2, explain the change. So when we talk about change and we talk about explain, first state what is the change. Second, explain why there's a change. Third, you elaborate why the change occurred. Okay, so the first thing, state. So if you look at the thing between time A and B, the change is the decrease in amount of oxygen. Okay, this refers to amount of oxygen. Okay, so there's a decrease in amount of oxygen. Why? Because oxygen is your final electron acceptor. See how all the answers, we are going back to the idea of how O2 is your final electron acceptor. Okay, so O2 is your final electron acceptor in your oxidative phosphorylation. So as ATP, sorry, this is wrong. As ADP, a substrate required for oxphos is added, it allows oxphos to occur, depleting the amount of oxygen. Because before that, ADP was not present. So if ADP is not present, oxidative phosphorylation cannot occur. If oxidative phosphorylation does not occur, no oxygen is used up. Okay, no oxygen is used up, therefore it remains constant. But because at time A, ADP was added, therefore afterwards your amount of oxygen decreased. Then lastly, account for the shape of the graph after time C. So after time C, it remained the same. So this is like your typical practical kind of question, right? typical enzyme kind of question as well. So the shape, so you have to identify your shape, explain why it is that shape. So the shape is that it's a straight horizontal line. And it is a straight horizontal line because your respiratory substrate has been completely depleted, completely used up. Okay, the keyword here is completely. If you don't use the word completely and just say it has been used up, it is left open to interpretation. So sometimes they might not give you the mark because they say, oh, there's still some left. Then they don't give you the mark. Completely is needed and the shape of your graph is needed. Okay,